I can ask one question for maybe Tanya and Anushree. In terms of the administration of combination therapy, in terms of timing and is that something you take into account in your screens at all? Because the current regimen is 4-4, right? 4 drugs, 2 months, 2 drugs, 4 months.
So in terms of TB, yeah, it's in the, so we have a target product profile, but we also have a target regimen profile. And it does add an extra layer of complication. At the early stages, you can't really look at that because you don't know what the dose will be. You don't know what the pharmacokinetics are like in the human. But certainly when you get to the later stages, you're really looking for things that are complementary. And for TB, ideally, it's a once a day therapy. So if you're looking at all 3 agents or 4 agents are going to be once a day, then hopefully that means that you can dose them at the same time. For us, obviously, there's another complication that we have to look very carefully at drug-drug interactions, and not just for the TB drugs, but for ART as well. There can be no drug-drug interactions for the HIV medicines or the TB medicines. So it kind of adds another layer of complexity at the later stages. And so you do build in some assays at the beginning to make sure that you're looking at those, but you can't really address it until you get the final kind of agents. But yeah, it's difficult.
All right. So for our therapeutic platforms, right now we are investigating, like when we do combinations, we are actually using them together. But that's not to say that timing of various antibiotics is not important. That's something that we have to investigate more. And it, again, as Tanya said, you have to look into the drug interactions, what kinds of interactions. You're getting synergy, you get antagonism, and both of them could be useful in the context of resistance, but engineering that is another layer. And that's something I think we should definitely do to build smarter regimens. But so we haven't done it yet because we're just focusing on building new therapies, but that's something definitely we would like to do once we have, once we kind of move this further ahead. That's something definitely we should explore.
I'll just ask one more follow-up before I go to Max. So in terms of, you discussed drug-drug interactions, but there's also host-induced changes in the pathogen and also drug-host interactions. And the pathogen can really take advantage of some of those interactions to gain tolerance and then be more prepared, even before you reach MIC levels in the tissue. So how does that affect your screens?
So it's a consideration at the later stage, but it's very hard to build those into screens when you're looking at hundreds of thousands or millions of compounds. I think there are a lot of things that you need to look at later on. And so as we start to go through the discovery process, as we get more progressive, into things that we think are going to succeed, then we'll start to build in other things. And it's very hard to predict with some of those. You can use the animal infection models, but you can't always predict what's going to happen in the human. And so at least for TB, the first efficacy trials will be using what we call EBA studies, or the early bactericidal activity studies. They are often done with the individual agent, but now people are starting to build those in to say, can we do the 2 agents together? So you actually see the outcome of the combination. You might not understand why you see antagonism or synergy, but you can at least measure what's happening in the human, because in the end, you're always going to be using a regimen therapy. But all of those things are considerations. They're just very hard technically to build into a screening platform, but you can certainly use them as you go along. And I wonder if I could follow up with my question, which was kind of unrelated, was I was really interested in HIV. So in TB, there's a lot of interest in combining drugs with vaccines or immunotherapeutics, the idea of the host-directed therapy. And I wondered whether that was a possibility for HIV.
Yes, it's really being looked at in 2 settings. One is therapeutically, vaccines that similar, many of those that I even talked about are being looked at for therapeutics in combination with antiretroviral strategies. In fact, we're also using pre-exposure prophylaxis, offering that to volunteers in our preventive HIV vaccine trials, since that has been shown to be effective in preventing infection. That does mean now that our studies are larger and going to take longer to do, and we're monitoring to see whether or not individuals are taking the pre-exposure prophylaxis. But I think that the combination types of interventions are really important to get there.
Max?
Yes. My questions are for Anushree. I was very impressed by the novelty of the approaches that you're taking. I have some questions about how large some of the challenges that remain are. Some of them that I'm concerned about. Well, I have like 3, but maybe I should ask one at a time. So with the PNAs. One of the big issues I see is the possibility of cross-reaction with things in the human genome or transcriptome, depending on what you target. How long are you going to have to make the PNAs in order to prevent that, and how much harder is that going to make the PNAs to get into the cell?
Yeah, that's very important. So, so the first thing that we are doing is in our bioinformatics toolbox, we are including, considering off-target effects within the human transcriptome. So to begin with, so the studies that we have done, we find that the PNA is most effective if you're targeting the upstream region, the 5' UTR. So that's a big cutoff itself, because a lot of the PNA targets and off-targets could possibly be between in the middle or 3' end of the transcript. So that'll triage a whole number of off-targets within the human transcriptome. Now let's consider if there are some off-targets within the 5' UTR of the human transcriptome. Again, in our informatics toolbox, we are assigning scores that will basically rank the PNAs, and the goal will obviously be minimizing impacts on the human transcriptome. Now, getting to your question about how long the PNA needs to be, scientists have designed PNAs as long as 20 to 30. And synthesis is fine. When it comes to transport, you have to use some other smarter ways. I was just discussing, using nanoparticles is one strategy which people are using. But also, when you make a longer PNA, your chances of off-target activity actually increase.
Okay.
So you have to see the break-even point over there. Now, the final point is also, like what we've thought about is, you have to think about the stoichiometric ratio between the mRNA and the PNA. And human transcriptomes, in general, have higher abundance compared to bacterial transcripts. So that is also something that we have to test, the relative ratio. And we do think there is enough window of opportunity for the PNA to be successful. We have run kind of toxicity experiments with the PNA with various cell lines, and we have gone as high as 80 micromolar, which is painful for me to do, but we've still done it, and we don't see, so far we haven't seen any toxicity effects.
Thank you. With the quantum dot, is cadmium telluride poisonous to humans?
It's toxic, of course it is toxic. And so one thing, so the thing that I did not say is we are actually building other kinds of quantum dots made out of FDA-approved materials like titanium dioxide or zinc oxide. And the idea there is to mimic the electronic behavior of this particular quantum dot and to be able to make those radicals. So superoxide is a very good one that we found. We are also making hydroxyl. Hydroxyl has some other issues, but it's still a great radical. Then we're trying to make nitric oxide. So that is definitely a major focus of our, moving things forward in our lab. But going back to the cadmium telluride, we are working at nanomolar ranges, and we have tested the quantum dots with various kinds of cell lines. So it's still in vitro. For example, for macrophages, we start seeing the first toxicity, significance at above, say, 300 nanomolar for macrophages. For osteoblasts, we see that at 600 nanomolar. But for bacteria, the quantum dots are effective as low as 10 to 35 nanomolar. So there is a window of opportunity. But again, we are very cognizant of the fact that we would avoid using cadmium telluride, even though we see it's more of the electronic behavior, not so much the material properties. But that's a major focus right now in our lab. So yeah, we are trying to address that.
Okay. And the third thing is, it seems to me that these approaches have some significant advantages. For example, with the PNAs, it doesn't seem, if you can deliver one, I don't see why you shouldn't be able to deliver a cocktail. So are there additional issues with concentration that come up when trying to combine those?
You mean like delivery issues, or?
You said you had to balance the stoichiometry of
No, so when I said stoichiometry, I mean to say PNA to mRNA ratio. So
right, but trying to deliver multiple PNAs, is it difficult? Do you run into a problem caused by trying to optimize that for multiple PNAs at the same time?
No. So far we haven't seen that. But what happens is, if anything, if you have multiple PNAs, each PNA has its own therapeutic effect. If anything, you have to decrease the dose of the PNAs because there is going to be synergy. So that is not a challenge. And if anything, eventually, the goal is to give cocktail therapy. So what we envision is, instead of having 4 capsules, create just 1 capsule that is basically 4 PNAs at once.
Thank you very much.
Can someone go wild and say that bacteria resistance is a developed countries problem and viruses and vaccine is a developing countries? Like, I was just thinking the cases where antibiotic resistance to the bacteria rise. How often is in the developing countries and how often is in developed?
Oh.
And how is this can influence the prevention, the prevention solution? So how often, for instance, and I'm thinking here mostly about clinics and the case of antibiotic-resistant bacteria in hospitals, if necessarily we need a pill or simply another prevention method.
Yeah. So these are 2 different things. So one is the rate of generation of antibiotic-resistant strains. So it is a function of antimicrobial stewardship in various countries, and as Marc was presenting today, your antibiotic resistance is also a function of antibiotic use. And if it's used, casually, then you'll have greater incidence of resistance. So I would say that right now in the developing countries where the stewardship is still being worked on, there is more source of generation of antibiotic resistance. That's the current thought. However, what I see is in today's time, nothing is local, everything is global, right? You cannot localize even resistance infections in one country, one location. People travel, bugs travel with us, and we don't even know about it. So I guess we have to figure out the solutions anyways, and whatever stewardship has to be applied truly on a global scale. Now, going to the prevention part of it, it's extremely important, but prevention can be much simpler than giving an antibiotic treatment, just making sure the surface is more sanitized. And hospitals are taking various kinds of measures, and in fact, recently we just started a collaboration with a major hospital in India, in AIIMS, and they are actually trying to really step up prevention methods, and there are even conversations about using, say, the quantum dot as a platform to aerosolize, to sanitize, in air microbes. So I think hospitals and at least a major number of countries are beginning to take this seriously, because it seems to be like a huge problem. But maybe Marc, you could add more to this.
Yeah, I think I agree with everything you said. I would maybe add that antibiotic use, including misuse, but not only misuse, is what selects for resistance. And what controls resistance, what's known to control resistance, is preventing disease in the first place, and infection control in hospitals and vaccine and other things. So it turns out that that developed countries are probably the only places in the world, a few developed countries, mostly in Northern Europe, are the almost the only places in the world where it's clear that antibiotic resistance is relatively under control. And every place can afford some antibiotics, because most antibiotics are generic and very cheap. So there's no place really that doesn't use antibiotics, and the places with the most control over it are the Netherlands and the Nordic countries. So I think certainly the New Delhi metallo-beta-lactamase that was mentioned is one example of something coming out of India, and the MCR1 out of China for colistin resistance is another recent headline example. So there are some examples of globalized resistance elements coming out of the developing world, but it is truly a global problem, and it's expensive to keep it under control.
So, I would just comment from the TB perspective. It's obviously the other way around, where, the big TB problem is in the developing countries. And so if you look at the US, the TB rate is very low. You still have a problem with drug resistance, but it's in the developing countries where you have the big problem with AMR and TB. But one other thing that I don't think has been touched on is there's a big issue with antibiotic persistence in the environment. So it's not just a case of when you're being treated, and there are certain areas, in Africa where there are a lot of antibiotics in the environment, and they're pretty stable. And I think that is something that, as kind of drug discoverers, we haven't really taken on board, is when you're trying to develop new antibacterials, we go for the thing that is the most stable. We want it to be stable under all conditions at all temperatures. And actually that, in some ways, could be counterproductive in terms of spreading resistance.
Need them compostable like the plates.
Yeah. Thank you.
Can I be controversial? Isn't the new frontier possibly nano scavenging, which is going on, I know, in research at Harvard? But actually, if you look at the literature, the 3 countries that are experimenting in nano scavenging, both for bacteria and viruses, is going on mainly in China, India, and Hong Kong. And might the message be, just like the big life science companies had agreed some time ago, that the small incremental improvements using antibacterial agents was really not worth the amount of money that was going into it, and therefore they had stopped producing antibiotics because they just didn't think it was a cost-effective approach. And just because Sally Davison persuaded Jim O'Neill to produce a hypothetical scenario of how AMR might create some great reductions in GDP, was possibly not a sensible approach in the longer term.
So I have to confess, maybe it was more of a mic issue. We couldn't hear the whole thing that clearly, but I guess I understood maybe parts of it. So I guess your initial question, so first question was, and correct me if I'm wrong, was on the nano scavenging part of it? Yeah, right. So I guess, you're right that one has to be responsible in terms of when you're working with nanomaterials and quantum dots and heavy metals and stuff, how much, like going back to the previous point, like you are releasing stuff in the environment, and then do you see that much therapeutic effect or not? Those are things obviously you want to consider very seriously when you're making a therapeutic. And there has been literature where people have used really high concentrations of quantum dots, like say micrograms, milligrams scale, which I think is a lot. And in those cases, we find that in those papers, there's a lot of material dependent effects. And so in our system, we have been very careful in terms of focusing on, the mechanism and focusing on, what exactly this quantum dot is doing, what kind of radical it's producing, and how do we engineer the quantum dot so that we only see therapeutic effect because of the radical produced, but not because of the material. And as a result, our therapeutic is very effective at nanomolar scale. Now, having said that, as I mentioned before, we are being very careful about the material we use, and that's one of the reasons why we have. We are going to start in vivo work, but one of the major things we want to do is to kind of shift to other kinds of materials that are FDA approved, that are body friendly, but mimic similar electronic behaviors. And that's a major emphasis and responsibility we take. But yeah, there has been a lot of let's explore various metals. And those, in fact, therapies are not that effective because you see they're using milligrams. That's way too much. So maybe the potential has sometimes been kind of shadowed, kind of hidden by maybe work which appears as if there is just like you're just exploring things. But I do see potential because if you think of these highly drug-resistant pathogens, many of them are thriving in even the last resort antibiotics. And for patients where nothing is working, you really have to start exploring new scenarios. So everything is perspective, everything is context. And if you have technologies which I do believe need to be further pushed and improved. And if sequencing can come this far, I believe we can definitely move therapies much further. And hopefully we have alternates that can treat these really nasty and smart pathogens at the same time.
Can I,
Please.
thank you. There's a theme that seems to me come across all of your presentations, which I really, by the way, think was superb. This whole, thank you. The whole discussion you've taken us through this today has just been beautiful. I'd like to connect it back, however, to where we started yesterday with global health issues. And this, I'm going to sound a little about the Pasteur-Beauchamp debate, right? We've been focusing on the bug here, and we've been focusing on therapeutics to the bug, but there's a host, right? And the host is the environment in which the bug lives. And that has to do with the ecology, which takes us to Lee Hood's talk about the nature of how we look at the whole system in terms of the ecology of this disease in either HIV or tuberculosis. So I'm wondering, when we're starting to do these studies, how are we looking at the environment in which the bug is embedded and the relationships of their immune system to things that are fairly simple about eating and about toxin exposure and about stress and about sleep and the nature of the environment, the microenvironment in which they're living. It seems to me that that's a variable that can have as big of impact upon the pathogenicity of the organism as the bug itself. I'd just like to know, how does that weave its way into the thinking as to how we look at the remediation of these global problems?
Yeah, well, certainly in terms of TB, there are a lot of other factors that play in as well. I mean, it's not just some of the ones you mentioned, but there's nutrition, and it's certainly clear that the biggest decrease in TB, at least in Europe and the developed world, was actually from improved living standards, not from drugs or vaccines. So I think there are different ways to solve some of the problems. Whether they're tractable, whether they're actually going to happen in the short to medium term is another question. I think from our perspective, what we're starting to understand is, even though we look at things from the bacterial perspective, and certainly with TB, it's an intracellular pathogen, or at least partly, we do consider the host in part when we're looking at that. But it's very clear that even with the standard antibiotics that you think work directly on the pathogen, actually within the human host, those antibiotics do work differently, and there is an immune component to some of them. So, and I think we're only scratching the surface of where that interaction occurs. But I think that's the move towards it. And then the idea that you can combine, as Gilla mentioned yesterday, the host-directed response, which is going to vary globally. If you can bring that into play together with something that's targeting the organism, I think that can be very powerful. I think for us, we're just scratching the surface. Maybe other areas are further developed than we are.
I could say something here. I think this is working. Yeah. It's a really good question. The way we're approaching it now for HIV vaccine development is to think about a whole host of baseline predictors. And then we hope, if there is funding, to really then be able to look at that at a more distant time point as well. But the list of baseline predictors is really growing, and it has many components of what you just mentioned. I don't think we've started thinking about how much sleep our volunteers are getting, but we are thinking about what they eat, their nutritional status, what else they've been exposed to in their lifetimes and currently, and the list is just very long. Ultimately, we really need to figure out what are going to be the critical components that are going to drive that immune response. But it's important, and it really helps us to also think about designing our studies, because we'd like to know, what might the placebos have responded, how might they have responded, that have that almost similar composition at baseline. So it's good.
Yeah, and I would just add that, as with TB, for all forms of infectious disease mortality, if you look at the curve over the 20th century, and for TB you can look back 300 years, but for other infectious diseases, as far as I know, you can see the 20th century. There's a spike in 1918 during a downward trend, and if you look for a bend in the curve to become more sharply downwards around 1950, as vaccines and drugs, antibiotics, you don't see it. It's just a continuation of a downward trend. So there's clearly a lot going on that's not about prevention by vaccines or treatment with antibiotics. Nevertheless, those clearly have also a big impact. In terms of drug resistance, one area that we've gotten very interested in is the sort of benefits of vaccines as an answer to the drug resistance problem. They've been sort of seen as separate worlds, and different people work on them and whatever, but vaccines modulate the immune system, and although some vaccines are incredibly challenging to create, like an HIV vaccine, others exist already, and it's a matter of deployment, like the pneumococcal vaccine, for example. And the estimates are that enormous amounts of antibiotic, first of all, that you just prevent disease, and if you don't have the disease, you don't care whether it would have been resistant. So the goal is to prevent disease, not to prevent resistance per se. And then beyond that, pneumococcal vaccines, and we imagine also a hypothetical RSV vaccine or flu vaccine, or existing or better flu vaccines would also prevent antibiotic use because people both inappropriately treat viral infections and also appropriately treat the bacterial consequences. So that's a very interesting way of using the host as a way to get to the pathogen.
So I'll ask one. Do you want to respond? No. So one last question is, I think it's probably more philosophical in terms of, I think, building on what was asked earlier, is at the end of the day, this is a systems problem. I mean, you have the heterogeneity at the level of the host. All of us are different. Heterogeneity at the level of the pathogen. There's spatiotemporal heterogeneity even within the same individual, even within the same granuloma. And the pathogen has mechanisms to do some bed hedging and hide away from the immune system from drug treatments. And the treatment itself induces the pathogen to become even more tolerant to the drug treatment. So given all of these complex interactions that are happening across multiple scales, do you think the systems we're using, well, I'm using this word system in a different way right now, is suitable, or are there major gaps in trying to understand how to find the right kind of drugs and how to deliver them most effectively?
I mean, I believe, yeah, I mean, there are certainly gaps, because I think this is more of an emerging consensus now in the field that there is obviously a lot of heterogeneity. And, as I mentioned at the beginning of my talk, you work with good old lab strains and they are evolving in a matter of days and being tolerant. And we work with intracellular infections and salmonella, and they're really adaptive because your host changes, they adapt very quickly. So I think, at least in our lab, we have come in terms to the fact that there is going to be variability, and it is to be expected. And so we are thinking the other way, which is, okay, how do we design therapy which can overcome, despite this variability, how do we defeat the bug? And I think that's the new paradigm, and that's something, there is a consensus. But to really have this as a therapy out there will be one of our goals for sure. But I mean, there is now appreciation for this, which is probably rather a new phenomena. And then there is also understanding that, like regimens of antibiotics, you want to cycle them, understanding how pathogens are evolving, whether synergistic antibiotics are good, are they selecting for resistance, or how should we combine antibiotics? So the people are coming from various perspectives. How should we interact with the host? How can the host, because host can be really helpful in clearing infection, and how do we boost the host response? And eventually, I think it's going to be coming from all directions to deal with these smart pathogens. But what we have found from our research is that even though they're really good, really smart, they can evolve, but probably there are certain weaknesses we can exploit. And some of our work shows that, and I think if we keep pushing that frontier, I do think we'll have good strategies in place. So,
Marc?
Yeah, maybe just to flip it around a little, I would highlight one success at the systems level, in my opinion, going back to the important omics of economics. One of the real tensions has been the notion that antibiotic discovery is disincentivized for various reasons, most importantly, that the best new antibiotic would be one that only one person had control over, that we would keep on the shelf, which would prevent recouping the costs. So there has been movement in the direction through CARB-X and other efforts of what they call delinkage, where the reimbursement for the drug is separated from the sales and from the consumption of the drug. So that I think is an important example of us identifying a systems barrier and at least trying to solve it. We'll see how well that works. I would also add just that one of the good things about vaccines is the notion that in that case, the economic incentives are aligned, because prevention, you sell a vaccine, it prevents the infection. So that's one nice thing about vaccines as part of this overall solution.
So, I mean, I would come back to the kind of lab approaches that we're taking. I think that we are starting to use the systems approaches to look at certainly in vitro bacterial systems and to try and understand the biology from a more holistic perspective. But I think where we see the biggest gap is translating that into the human setting. So we don't have a lot of biomarkers for disease cure, for disease development, for transition from latent to active disease in TB. And we don't have the really predictive animal models that allow you to do the more sophisticated modeling. So I think for us, I see that's where our real gap is. Like, how do you translate what you do in vitro or in simple co-culture models into something that's going to be relevant to human infection?
I think in the case of vaccines, we are making progress to be able to look at humans in a more systematic way, immunologically. And it's important to do, and I think we're making strides in that. Where I think the difficulty lies in trying to then translate those approaches into pediatric settings, because it's just really hard to do it, and with small sample size, and so efforts need to go there to try to understand that evolving immature to mature immune system, and how can we really interrogate that. The other thing I think that just still stands as a major problem is decision making about what types of vaccines are going to get developed, and who are they for, and really coming up with sound approaches to move these things forward more quickly. And I think that it's really hard to take a really novel observation and see how that can play out as a vaccine without taking just so long. And particularly if it's for a pathogen that is more of a global health type pathogen to prevent, that a company is not going to be that interested in making. So.
Well, thank you to all the speakers. Let's give them one round of applause.